Welcome to Inert Chemistry. In today's video lecture, we are discussing about eukaryotic chromosomes. What are chromosomes? Chromosomes are long thread-like structures that are made of DNA and proteins. They are found in the nucleus of cells. They carry genes and contains instruction for the development, functioning and reproduction of living organisms. Different organisms have different number of chromosomes. Humans typically have 46 chromosomes that are organized into 23. A typical chromosome consists of various parts. The most important are centromere. This is actually the region where two sister chromatids are held together and where spindle fibers attach during the cell division. Here you see that this is the central point where that is centromere. Telomere. It is found at the end of chromosomes and telomeres protect your DNA from deterioration or fusion with other chromosomes. Here you see that these yellowish structures are telomeres. Chromatids. Chromosomes have two identical sister chromatids that are joined at the centromere. Here you see that these are the sister chromatids and this is the basic structure of a chromosome or of eukaryotic chromosome. Chemical composition of chromosomes. If we talk about the chemical composition of chromosomes, then a chromosome, it is made up of two things. The first is DNA and the second is proteins. The most abundant protein that is present in chromosomes is histone protein and the second part is DNA. Here you see that chromosome is made up of DNA and proteins. Although a small quantity of RNA is formed, but its percentage is very less, so we can neglect it. But mostly it is made up of DNA and proteins. Proteins that are present in DNA are histone proteins, mostly, but some other proteins are also present. And the DNA, it is deoxyribonucleic acid. It is made up of phosphoric acid, deoxyribose sugar and nitrogenous bases. And the nitrogenous bases are further of four types: adenine, guanine, cytosine and thymine. If you want to know about the structure of DNA, we will make another video for this. Types of eukaryotic chromosomes. In eukaryotic chromosome, there are two types: autosomes and sex chromosomes. Autosomes are the chromosomes that determine most of organisms' traits, excluding the sex determination. While sex chromosomes are those chromosomes that are involved in individuals' sex determination. In case of female, they are XX, while in case of male, they are XY. According to the number of centromere, the eukaryotic chromosomes are divided into acentric, monocentric, dicentric, and polycentric. Acentric are those in that are without any centromere, monocentric with one centromere, dicentric with two centromeres, and polycentric with more than two centromeres. And according to the position of the centromere, the eukaryotic chromosomes are divided as follows. Metacentric. The metacentric chromosomes are V-shaped. V-shaped. Centromere occur in the center and forming two equal arms. This first is a metacentric chromosome. The second is submetacentric. The submetacentric chromosomes are J-shaped or L-shaped. In these, the centromere occur near the center or at the medium portion of the chromosome and thus forming two unequal arms. This second chromosome here shown, it is submetacentric. Acrocentric. The acrocentric chromosomes are also rod-like in shape, but these have centromere at one end. and thus giving a very short arm and an exceptionally long arm. Telocentric. The rod-like chromosomes which have the centromere on the proximal end, means at the end, are known as telocentric chromosomes, and they are just like eye shape. Chromosome organization in eukaryotic cells. During the G1 phase of the cell cycle, there is only a single chromosome, means just like this, there is only a single chromosome. But as phase chromosome duplicate to two sister chromatids, means this chromosome duplicate and there are two sister chromatids that are attached to the same centromere. During the G2 phase, the chromosome is also in same shape, means in the form of two sister chromatids that are joined by centromere. During the M phase, these centromeres separate from each other and daughter chromosomes are formed from sister chromatids. As you seen here, this is the chromosome, and after the mitotic division or mitosis or meiosis, it is divided and it is changed into two sister chromatids. Here we see the chromatin structure. What is chromatin? Chromatin, it is made up of DNA and protein. The DNA is deoxyribonucleic acid, and the proteins that are present, they are of two types, histones and non-histones. Both the proteins are necessary for physical structure of chromosome, means both histone and non-histone proteins are necessary for physical and structure of your chromosome. First of all, we will take a bit look on histones. The most abundant proteins that are present in DNA are histone proteins. These are small proteins that are basic in nature, and these proteins contain large quantity of arginine and lysine amino acids. Due to the presence of these arginine and lysine amino acids, these are positively charged. Due to the presence of these arginine and lysine, the proteins get positive charge and thus they bind to negatively charged DNA. There are 5 types of histone proteins: H1, H2A, H2B, H3 and H4. The amount of histones is equal to the amount of DNA in a chromatin. Non-histone proteins. All the proteins that are associated with DNA except the histone proteins are called non-histone proteins. For example, the proteins that bind to DNA during its replication, repair, transcription, gene regulation or recombination, these are called non-histone proteins. They are mainly negatively charged and hence they bind to positive histone proteins. Here you see that the organization of eukaryotic chromosomes. Here you see some key differences between prokaryotic chromosome and eukaryotic chromosome. Thanks for watching the video. We will soon come with another video. Kindly like, share and subscribe to the channel.